Welcome to today's Explainer. We're transforming cutting-edge botanical research into a journey through the high-tech, glass-enclosed future of plant science. And it's a future that might just save our most vital medicines. So do you know where your medicines, fragrances, and even your cosmetics actually come from? We all kind of tend to picture these vast, untouched natural landscapes, right? Maybe serene farmers gently harvesting herbs at dawn. But the modern reality of where these crucial ingredients are sourced is far more complex, and frankly, it's pretty urgent. The shocking reality we found in our sources is that the pharmaceutical and cosmetic industries are essentially loving nature to death, driven by massive global population growth and a skyrocketing reliance on natural remedies. We're systematically over-harvesting valued medicinal and aromatic plants right out of the wild. This relentless, totally unsustainable extraction is pushing vital, life-saving plant species completely to the brink of extinction. Here's our roadmap for today's Explainer. We'll start with the medicinal plant crisis, step inside the glass factory, and formulate the perfect plant diet. Then, we'll explore high-tech plant conservation, look at some real-world impacts in Pakistan, and finally, cast a vision for the future. Let's kick things off with Section 1, the medicinal plant crisis. We're facing massive, interconnected global hurdles when it comes to relying on traditional agriculture. I'm talking about severe overharvesting from wild populations, incredibly low and unpredictable yields of the actual medicinal compounds we need, painfully slow plant growth cycles, and devastating habitat loss driven by a changing climate. But the good news? Biotechnology provides direct, elegant answers to every single one of these problems. We can actually counter overharvesting by producing enormous amounts of planting material from a single tiny plant cutting, a concept known as an explant. This micropropagation process allows us to scale up incredibly fast. We can also beat low yields by carefully optimizing laboratory conditions, which often results in way more medicine being produced than what the mother plant could ever yield in nature. Plus, controlled indoor environments completely bypass those slow natural growth seasons, and we can fight habitat loss through targeted high-tech conservation methods that keep these species safe indefinitely. Moving on to Section 2, inside the glass factory. The absolute crucial concept unlocking all of this potential is in vitro cultivation, which literally translates from Latin as in glass. Just imagine creating an entirely sterile, highly controlled cellular factory right inside a laboratory beaker or test tube. By isolating specific plant cells, tissues, or organs and placing them in a totally aseptic environment, scientists completely eliminate the threats of pests, diseases, and unpredictable weather. And by rigorously controlling every single variable, from the light spectrum all the way down to the temperature, these lab-grown cells can be coaxed into absolute hyperdrive. They actually synthesize and accumulate higher amounts of highly prized secondary metabolites. Those are the chemical compounds we use for drugs. And they do this way better than the original plant ever managed to produce while struggling to survive out in the wild. Now, let's talk about Section 3, the perfect plant diet. Because you really can't just throw some dirt in a sterile beaker and hope for the best, right? The success of these intricate cell factories depends entirely on a precise nutritional diet called the culture medium, with the most famous baseline recipe being the 1962 Murshig and Skoog medium. Think of this as a highly calibrated IV drip for a high-performance athlete, but designed entirely for a plant. These isolated cells are extremely demanding. They need macronutrients like nitrogen and potassium for their basic building blocks, and micronutrients like iron and zinc for cellular function. Because they usually aren't photosynthesizing efficiently yet, they need a direct carbon source, which is typically a 2 to 3% sucrose mix for energy. On top of that, scientists carefully blend in specific plant growth regulators, vitamins, and amino acids to direct the cell's exact behavior. Finally, to give the cells a physical, semi-solid structure to sit and grow on, they use a gelling agent, typically 0.8 to 1% agar. It's essentially highly nutritious plant jello. That brings us to Section 4, high-tech plant conservation. Beyond just mass-producing medicine, we can use this exact same technology to save rare, endangered, and threatened species from vanishing forever. And this operates through 3 really fascinating high-tech steps. Step 1 is slow growth storage. By slightly reducing temperatures or adding osmotic regulators, which are essentially chemical agents that control the plant's water balance, scientists can literally hit the brakes on a plant's metabolism. This dramatically extends the time a plant can safely sit on a lab shelf without needing fresh nutrients. Step 2 takes preservation to the absolute extreme. cryopreservation. Here, they submerge the biological material in liquid nitrogen at a freezing -196 degrees Celsius. This completely arrests all metabolic and cellular activity, theoretically allowing for unlimited, centuries-long storage with virtually zero risk of genetic mutation. And step 3 is truly sci-fi. It's synthetic seeds. Researchers take lab-grown plant tissue called somatic embryos and encapsulate them inside protective, nutrient-rich alginate beads. This creates artificial, lab-made seeds that can be easily shipped across the globe and directly sown into soil, just like conventional seeds, but without ever needing a mother plant to produce them. Let's ground this in reality with Section 5, Real-World Impact in Pakistan. These in vitro solutions are actively moving out of the theoretical realm and rescuing endangered species in Pakistan right now. For example, take the Himalayan mayapple. It's deeply endangered in the wild, yet it remains a primary biological source for podophyllitoxin, a compound that is absolutely vital for manufacturing anti-cancer drugs. To prevent its extinction while maintaining the drug supply, researchers in Pakistan developed a successful slow-growth storage protocol at exactly 5 degrees Celsius, using very specific additives like sorbitol to preserve this critical species. Meanwhile, other teams are using callus cultures. These are essentially masses of unspecialized stem cell-like plant tissue that can grow incredibly fast in a lab. They're using them to rescue and rapidly multiply medicinal plants from the Cholistan desert that are quickly vanishing due to severe overgrazing and climate shifts. But get this, the most staggering real-world breakthrough in this field is a massive 97.75% cost reduction in production. Now, how is a discount that huge even possible in high-tech biotechnology? Well, one of the biggest bottleneck expenses in a commercial tissue culture lab is lab-grade agar, that gelatinous foundation we discussed earlier. Researchers at the University of Swat, who were studying the famous sweet herb stevia, achieved this massive cost reduction simply by walking down the grocery aisle. They successfully swapped out the highly expensive laboratory agar for everyday commercially available semolina. They even tested sago pudding mix, which dropped costs by over 90%. It's a brilliant, almost deeply funny example of human ingenuity, using low-cost, local baking alternatives to make high-tech conservation economically viable on a massive scale. And that leads us to our final stop, Section 6, a vision for the future. The vision outline for Pakistan, and the blueprint it really offers for the rest of the world, is sweeping and highly integrated. The future of natural medicine relies heavily on building vast interconnected networks of regional tissue culture facilities that can rapidly multiply locally threatened species. It means officially weaving these specific in vitro conservation techniques directly into national and global biodiversity action plans, moving them from niche science into standard environmental policy. It also calls for powerful public-private partnerships. Governments and private medical corporations simply must team up to fund the industrial-scale extraction of high-value compounds. Add in comprehensive training programs for a whole new generation of biotechnologists, open international borders for sharing cryopreserved samples, and ultimately the establishment of massive bioreactor facilities. I want you to think of giant, sterile, multi-story stainless steel vats, silently churning out life-saving pharmaceuticals and preserving ancient plant genetics, all grown safely inside glass. As we wrap up this explainer, I want to leave you with this provocative thought based on everything we've uncovered today. If our natural world continues to shrink at its current pace, will our future forests and our future pharmacies exist entirely in glass? It's an absolute incredible testament to human innovation that we can decode, nurture, and preserve complex botanical life inside a perfectly sterile lab. But it should also serve as a stark, lingering reminder of just how fragile the ecosystems are outside those glass walls. We have the technology to save nature's medicine cabinet, but the real test is how we choose to apply it. Thanks for joining this explainer, and keep exploring the fascinating ideas shaping our future.